Good morning, everybody. Let us see about photorespirations. From this heading itself, we can easily understand its definition. That is, excess respiration in green plants in the presence of light is called photorespiration. It was discovered by Dectet and he named this process as C2 cycle because two carbon group is involved in this. Respiration takes place both in dark and light. The light dependent is photorespiration and the light independent is dark respiration. The photorespiration is 3 to 5 times greater than that of dark respiration. Three cell organelles are involved in photorespiration. They are chloroplast, peroxisome and mitochondria. These photorespiration is present in cotton, pulses, capsicum, peas, tomato, etc. and it is absent in grasses. Now let us see about its mechanism. Here 1,5-diphosphate is produced during photosynthesis with excess oxygen and low CO2 and it splits into 2-phosphoglycolic acid and 3-phosphoglyceric acid. The 3-phosphoglyceric acid enters the Calvin cycle, whereas from 2-phosphoglycolic acid the phosphate group is removed by an enzyme phosphatase so as to form glycolic acid. From the 2-phosphoglycolic acid, the phosphate group is removed. Then the glycolic acid is remained, and this glycolic acid enters into the peroxisome where it combines with oxygen. We already know that addition of oxygen is an oxidation process. So photorespiration is an oxidation process. The glycolic acid combines with oxygen and it forms glyoxylic acid and hydrogen peroxide. The hydrogen peroxide is toxic to plants, so it splits into water and oxygen. And this glyoxylic acid combines with amino acid and it forms glycine. And this glycine enters into the mitochondria from the peroxisome. In mitochondria, two glycine group condensed together to form an serine. From this serine, the amino group and the carbon dioxide liberated and it forms hydroxypyruvic acid, and this hydroxypyruvic acid undergoes reduction with NADH and it forms glycerate, and these glycerate undergoes phosphorylation in the presence of ATP and ADP and it forms 3-phosphoglycerate. So from this we know from 2-phosphoglycolic acid several processes were undertaken and finally it forms 3-phosphoglycerate. Remember that photorespiration is an oxidation process. Let us see about significance of photorespiration. The photorespiration helps in classifying the plants. It is generally present in C3 plants and it is absent in C4 plants. It protects the plants from photooxidative damage. Photorespiration uses energy in the form of ATP and reduced nucleotides, whereas the normal respiration yields ATP and reduced nucleotides. It is believed that photorespiration was common in earlier days when CO2 content was too low to allow higher rates. Let us see about respiration. Respiration is the reverse process of photosynthesis. In this, the carbohydrate is break down into water and CO2. Let us see about in respiration, the oxidation of variable organisms takes place in which glucose is common. It is not an easy step. It undergoes many steps which results in the release of energy in the form of ATP molecules and also forms number of carbon compounds. We already know that respiration is of two types. One is aerobic respiration and another one is anaerobic respiration. Aerobic respiration, which takes place in the presence of oxygen and the respiratory substrate completely oxidized and carbon dioxide water are formed as an end products. Here let us see the reaction for aerobic respiration, that is glucose plus oxygen gives CO2, that is carbon dioxide plus water plus some energy. It occurs in very common manner. Next, anaerobic respiration or fermentation. It takes place in the absence of oxygen and the respiratory substrate is incompletely oxidized. In addition to carbon dioxide, other components are also formed. It occurs very rare, but in yeast it is a common process. Here, glucose is converted into ethanol plus carbon dioxide and plus some energy. Let us see the difference between aerobic and fermentation. It occurs in all living cells of the plants throughout day and night. It occurs outside the plant cells and certain microorganisms. presence of oxygen, and it takes place in absence of oxygen. N products are CO2 and H2O. N products are CO2 and alcohol or other organic acid. It is non-toxic to plants. It is toxic to plants. Complete oxidation of food material is observed. Incomplete oxidation is observed. Respiratory quotient, it is the ratio of volume of CO2 evolved to the volume of O2 consumed. This RQ value depends on the respiratory substrate used during photosynthesis. When carbohydrate is the respiratory substrate, then the RQ value will be 1 or unity. Let us easily understand this by this equation. Here glucose plus oxygen gives CO2 plus H2O. Here the volume of CO2 is 6 and the volume of O2 is 6. So 6 by 6 is equal to 1 or unity. When fats are the respiratory substrate, the RQ will be less than 1 because they contain more oxygen than CO2. So when we divide 102 by 145, we get 0.7, which is less than 1. When organic acids are their respiratory substrate, the RQ value will be more than 1 because here the carbon dioxide volume is greater than that of oxygen volume. So 4 by 3 is equal to 1.3, which is greater than 1. Factors affecting respiration. There are two factors. One is external and internal factors. Let us see about it. External factors. First one is temperature. Temperature influences in the rate of respirations. The optimum temperature is 30 degrees Celsius, in which the rate of respiration is high. The minimum temperature is 0 degrees Celsius, in which the enzymes becomes inactive. So only we are keeping fruits and vegetables in cool place or a refrigerator. Maximum temperature is 30 degrees Celsius, in which the respiration rate greatly decreases or even stops due to the denaturation of enzymes. Next one is oxygen. If the oxygen content is high, then the rate of respiration is also high. In the complete absence of oxygen, anaerobic respiration takes place, which produces alcohol that are toxic to the plants. In this some availability of oxygen, then the aerobic respiration starts. When the aerobic respiration is at optimum level, then the anaerobic respiration stops. This is known as extinction point. Next is carbon dioxide. If the carbon dioxide content is high in poorly aerated soil, then it has a retarding effect on the rate of respiration. Next one is inorganic salts. If the plants or tissues are transformed from the water to salt solution, then the rate of respiration increases. This process is called as salt respirations. Next is water. If there is a medium, then the respiratory enzymes become active. In the absence of medium, the respiratory enzymes become inactive. So here water acts as a medium. In the presence of water, the rate of reaction increases. In the absence of water, the rate of reaction decreases because water acts as a medium for the respiratory enzymes. Light, it has indirect effect on the rate of respirations through the synthesis of organic food matter in the process of photosynthesis. Wound or injury. This wound or injury increases the rate of respirations because the meristematic cells requires more energy to overcome or heal this wound or injury. When it requires some energy, the rate of respiration increases. So the wound or injury increases the rate of respirations. The amount of protoplasm and its rate of activity influences the rate of reactions. The rate of reaction is higher in young meristematic cells because it have more amount of protoplasm and no vacuoles. And the rate of respiration is lower in older mature leaves because of lesser amount of active protoplasm. Second one is concentration of respiratory substrate. If this concentration increases, then the rate of respiration also increases. Under starvation conditions, that is etiolated leaves, the rate of respiration slows down. If such etiolated leaves are supplied with sucrose solution for few days, even in darker conditions, then the rate of respiration increases. Let us see the difference between photorespiration and dark respiration. This dark respiration is also known as mitochondrial respiration. Photorespiration occurs in the presence of light. Dark respiration occurs in presence of both light and dark. Here, in photorespiration, the substrate is glycolate, and here the substrate may be carbohydrate, fat, or protein. It occurs in chloroplast, mitochondria, and peroxisome. The process occurs in cytoplasm and mitochondria. It occurs in temperate plant crops like wheat and cotton, mainly in C3 plants. It occurs in C4 plants such as maize and sugarcane. It occurs in green tissues of plant. It occurs in both green and non-green. The optimum temperature is 25 to 35 degrees Celsius. It is not temperature sensitivity. This increases with increase in CO2 concentration. Here, the process saturate at 2 to 3% of O2. If the O2 concentration increases beyond this, then there is no increase. Hydrogen peroxide is formed during reaction. Hydrogen peroxide is not formed. ATP molecules are not produced. Several ATP molecules are produced. Reduced coenzymes such as NADPH2, NADH2, FAH2 are not produced. Such enzymes are produced in dark respiration. One molecule of ammonia is released, no ammonia is produced. Thank you.